As people are coming in, I see lots of people logging in to join the conversation. I just want to say welcome. We're going to give just a couple minutes for people to join, and we'll get started here in just a couple. But again, thanks for joining us today for this really exciting conversation. And just sit tight for maybe just 1 minute while we make sure everybody has a chance to join. Okay, it looks like usually when we open this up, we get, just a whole roll of people who jump right in. So I wanted to make sure we caught all those before we got going. So given that, looks like we're ready to start. So welcome to this very exciting webinar, Exploration of a Novel Biomarker and the Connective Tissues Community. We're so grateful to get to be a part of this study and to offer it as a wonderful discussion place and point for asking questions to the community and any clinicians that are joining us today. Just a few little housekeeping things before we get started. Wanted to make sure that we always want to share the Help and Resource Center link. If you have any questions about this topic or any topic, whether it be physical health, mental health, how to work with your providers, anything related to living with Loeys, Dietz, Marfan, VEDS, or related condition, we really encourage you to go to marfan.org/ask. There's a form there, also a phone number that you can call or submit your information online, and our on-staff nurse, Jan, will get back to you just as soon as she can. She's wonderful, and if you have an opportunity to ask her any questions, we very much encourage it. Do never hesitate to reach out to Jan. Also wanted to throw out here that the topic we're talking about today, there is an infographic available that summarizes a little bit about what is a biomarker, why is it important, and what we hope to see from this study at loeysdietz.org/biomarker. And there is no hyphen in that. It's all one. It should just be Loeys Dietz, all one word. Sorry about that. It's the habit of typing it as the hyphenated. And then the last point I want to mention is that coming up in a month, just about a month, we have another webinar that may be of interest to folks who are on this one. It's just genetics, update on genetics research. It'll actually be Dr. Dietz that is presenting that topic at 7 p.m. on February 27th on Eastern time. You can register for that at marfan.org/calendar/genetics-research. And that is also available if you go to any of our division or Marfan websites too. If you just look for it on the calendar, it'll be there. Given all that, please help me welcome. I'm excited to have both Dr. Bart Loeys and Aline Verstraeten, close maybe, both from the University of Antwerp, presenting on this topic, both researching on the exploration of a novel biomarker for the connective tissues community. Just also want to throw a quick shout out to the Loeys community who had raised funds for research in this area, research that would affect the Loeys community and by extension, Marfan and other connective tissue communities. And those funds were used to help support this research. And we are frankly honored to get to do that. So with that, I'm going to turn it over to our presenters. And let me stop my share and hand it over. As they get set up too, if you have questions that you would like to submit, you can drop them in the Q&A. We'll monitor that closely for questions. If you do it in the chat, sometimes it's hard for us to keep track of where all they are. So if you do have a question, drop it in the Q&A and we'll do our best to get those answered in the course of the discussion. So with that, I'll hand it over.
Okay. Good evening, everybody. I hope everybody can hear us. Yes, okay. Thank you, Angela, for the very kind introduction. I also want, also on behalf of Aline, thank the Loeys Dietz Foundation for supporting this study on the exploration of a novel biomarker, which might hopefully not only be relevant to the Loeys Dietz patients, but also to the whole connective tissue community. Aline is a excellent colleague with a large track record also now in connective tissue research. And the way we designed this webinar is that I will try to give some general explanation on what a biomarker is, what good characteristics of an ideal biomarker would be, go a little bit into depth of those characteristics, and then Aline will address how we are trying to build on some preliminary findings and explore the possibility of a novel biomarker. So in the next hour, I will define a biomarker. We will talk about different types of biomarkers that are known, what key characteristics of a good biomarker are, how biomarkers can be used, specifically in the connective tissue field, which ones are currently known, and then Aline will talk about the process of identifying a novel one in Marfan and Loeys Dietz syndrome. So first of all, I would like to start by defining a biomarker. A biomarker is a biological marker that is a measurable indicator that can tell us something about a person's health or disease state, and that can be at different levels. For example, it can be a disease process in the body or a disease stage. So if we apply it to connective tissue disease, most relevant might be, for example, aortic root or ascending aortic aneurysm and its development. But it can also apply to other biological processes and can even be applied, and we'll give some examples of that to a person's response to a treatment or a medicine. So if we would have a medication that would make aneurysm better, then we could use the biomarker to see if it also predicts the outcome of the treatment or can tell us about the effect of the treatment. So as I said, in the connective tissue field, I think it's most relevant to tell us something about the severity of an aortic aneurysm, and especially when there is a risk for aortic dissection. So here is what we try to develop. We know that in most cases, a dissection is preceded by aneurysm development. And it would be ideal if we can find something that can help us to predict the evolution and can tell us at what time there is an imminent risk of aortic dissection, or if a dissection happened or there's a rupture. And we already know that there is some risk factors that are well known. Males have higher risk than females. We know that there is some environmental or lifestyle factors that influence the risk, such as hypertension, atherosclerosis. And we also know that genetic factors, and I will come back to that also to some degree, explain some variability. But I think we need to know more, and we need to know better how to try to help predict these catastrophic events. So if you look in the literature, you could imagine different types of biomarkers. The ones that first come to mind are molecules that could be detected in any body fluid. Most of the time it's blood, and that can be at different levels. It could be at the DNA level, but as you know, to make a protein, you have an intermediate that's called RNA, which is basically the messenger that tells the cell how to make a protein from the DNA. But also these proteins that are made in the body are metabolized, and also sometimes these metabolites are used as biomarkers. Another type of biomarkers could be more at the tissue level, and in this case I applied it to the aorta. We could look at specific characteristics of the tissue. Is there specific cells that appear in the aortic wall that go along with aneurysm or predict aneurysm type to be going into direction of dissection? Or is there any tissue characteristics that could help us to predict all that? And then the last one is imaging. And you all know that there is different types of imaging. You can do echoes, but in the biomarker world, most of the time, a CT scan or a specific type of scan, which is a PET scan, can be used. For example, inflammation can be detected, or magnetic resonance imaging can also be used. And we'll give some examples of that later on in the presentation. So if we think about the ideal biomarker or a good biomarker, I think we want it to be a simple test. For example, when we can do it with a simple blood test, it's easier than to do it with a magnetic resonance scan, which requires more equipment. And it preferably would also be an inexpensive test, because you want to do it throughout the lifetime. So you probably have to do it multiple times. So you don't want the test to be very expensive if you have to repeat it every couple of months or every couple of years. We also prefer the biomarker to be safe, and especially to be non-invasive. When I was talking on the last slide about tissue samples, we don't want to do invasive procedures where we have to take biopsies of the aorta to predict the evolution of an aneurysm. So that's not the type of biomarker that we would prefer. And then there is a couple of characteristics that have more to do with positive and negative predictive value. And I will explain that a little bit more in detail, because I think it's important to understand what kind of testing is done before a biomarker is really validated as a good biomarker. So I will try to explain that with some simple graphics. When we look at a disease state or disease progression, people can be largely divided into two groups. Either you're sick, you have the disease, or you're well, you don't have the disease. And we'll present that here with a sad face and a smiley face. And then if you do a test, any of the previous types that I discussed, you can have a test that is positive. It sounds a little bit strange, but that's how we use it in medicine. It means that the patient has the disease, so it might not be positive for the patient, but the test is positive. Or a test can be negative, which means that the patient does not have the disease and that the patient is well. So when we do a test now for either a healthy person or a diseased person, there is 4 possible test outcomes. And there's 2 that are right, which we call either a true positive or a true negative. So a true positive means that the patient is really sick and that the test comes back positive. A true negative means the patient is well and the test comes back negative. But there is also two scenarios in which the test outcome is really wrong. The first one is the false negative, which means that the patient is sick, but that the test comes back negative, so it doesn't tell us the truth. Or it's a false positive, the patient is really well, and the test comes back positive. And these are really very important characteristics on which we can do some calculations. And the first one that we look at is what the sensitivity of a test is, which means that we want to calculate how often a test is positive when a patient has a disease. And as you can see here in the graph, there is 5 sick people. They all have the test and it comes back positive 4 times, but 1 time it comes back negative. So in that case, the test is false negative. So in this case, the test has a sensitivity of 80%. Similar is something it's called specificity, and that tells you how often a test is negative when the patient is well. So here you see 5 healthy people, 4 of them have a negative test, 1 of them has a positive test. That's really a false positive because in reality there is no aneurysm, the patient is well. And it gives you a specificity of 80%. So the specificity tells you how good the test is at identifying the healthy or the well people. And then there's 2 other things that we calculate, is that we also want to know when a test comes back positive, how often it really tells you that the patient is sick. So in this case, we see 5 individuals that have a positive test. 4 of them are sick and 1 of them is not sick. So it means that the positive predictive value of the test is 80%. So it tells you after the test has been done, how likely it is that the patient will be sick. And the opposite of that is the negative predictive value, which doesn't tell you about the sick people, but about the people that are healthy, that are well. It's very similar. We want to know how often when a test is negative, the patient really does not have the disease. So after the test comes back negative, it will tell you how likely the patient is to be well. So when you bring this all together, this is the scenarios that we talked about. You have in green the ones that are true, the true positives, subjects with the disease that have a positive test, or the true negatives, subjects without the disease that don't have the disease. And then you can calculate the different values. And these values are very important because, of course, we want all of them to be as high as possible. We want the test to be sensitive in order to detect all the people that have the disease, but we also want it to be specific. So we don't want to make people to be told to be diseased if they in reality don't have the disease. And we can similarly calculate the positive and the negative predictive value. In addition to that, we also want, when we do the test in the lab, there is some technical requirements for the test. And we want the test to be accurate, correct, but we also want it to be precise, and it means consistent. So you can see in the graphics what is meant with that. So if it's accurate, it's really is predicting it always in a correct way, but it can be very precise, but not be accurate, as you can see in the second graph. So we want a combination of both. So your test will not show too much variability, and it always needs to detect when the disease is there. So these are some background informations on how to look at the validity or the characteristics of a biomarker. So why do we need this in connective tissue disease? You can say, oh, maybe we already have all the risk factors. We know when there will be a dissection. And in current practice, we indeed use the absolute or the relative aortic root size. So the absolute one would be the one in centimeters or in millimeters. The relative one would be the one normalized to the body size, so taking into account your gender, height, and weight. So that's really an important one. We do know that. We also look at the speed of progression. The faster it evolves, the higher probably the risk will be that there will be a dissection. And we also take into account a family history, because if one of your family members dissected, there is also a higher chance that you will experience a dissection. But unfortunately, these risk factors do not always predict the risk for aortic dissection very accurately. For example, if we would look at a cutoff of 5.5 centimeters, which would be considered as a very high risk of dissection, only small proportions of patients fulfill that. So the size and the progression rate by themselves do not always predict the risk for dissection in an accurate way. So there is a clear need to do better and to identify other factors. And one of the possibilities to do that is biomarkers. So then what could we do with those biomarkers? First of all, we could try to monitor the progression of aortic aneurysm diameter. So in the ideal situation, if the biomarker that you would look for example, in the blood, let's say we identified a molecule, you want the amount that's present in the blood to increase also with the progression of aortic diameter. So then we can use the value of that biomarker as a marker for progression of the aneurysm. In the ideal situation, it would even further increase if there is an imminent risk of dissection. So if we would do the blood sampling and we see a threefold increase rather than just a gradual increase, then it might tell us, oh, this patient is at risk for dissection. So then it can be used as a predictor for dissection. Alternatively, we can also sometimes use it to know when there is a dissection. So that would help, for example, in the emergency room when a patient comes in and has chest pain. Rather than doing more complex imaging, we can just take a blood sample and see if the biomarker indicates that a dissection is present. So that would be another possible application of identifying a biomarker. And for all these scenarios, we need to test its validity based on the characteristics that we just described. Another potential application would be even after a patient had surgery. We all know that there is a residual risk of aneurysm development in the parts of the aorta that were not that did not undergo surgery. So there again, it could help us to predict that. In medicine, biomarkers are also often used to monitor response to treatment. So if we see a decrease of the biomarker, again, it might indicate that the patient is responding well to the treatment, and it can help us to predict potentially also a later stabilizing of the aortic measurements. But if the biomarker can do that faster, it will help us to identify quicker which treatments are beneficial to which patient. And sometimes that's also used in clinical trial. If you have a very good biomarker that could tell you in advance that the aortic root will not continue to progress to dilate by a decrease of your biomarker, then it could also speed up clinical trials. So we don't have to wait 3 years to see the effect of the medication on the aortic size by doing the imaging, but we could learn it faster by seeing a decrease in a specific biomarker. So these are all potential applications that biomarkers can be used in aneurysm follow-up in connective tissue diseases. So how could it look like in connective tissue diseases? I will give some examples of the different categories that we talked about. So at the DNA level, at the imaging level, at the tissue level, metabolomics and proteomics. So a first example of something that could be integrated as a potential biomarker, and we already do that to some extent, is that if we can identify the primary cause of the mutation, that can help us to determine the cutoff that we need for surgery. This is not a perfect measure, and unfortunately, we also don't identify the primary mutation in every patient yet. So at present, if you look at all the patients with thoracic aortic aneurysm, and then probably we identify a genetic cause in 30%. So we cannot use it in every patient. So it's not a perfect predictor. Also, what we do observe is that even within a family, which is supposed to have the same genetic predisposition, that there is some variability, that one person with the same genetic mutation does present with the aortic aneurysm dissection, whereas a sibling, for example, with the same genetic change does not present it. So there is other factors that influence this primary mutation. And looking into that is one of the fields that could also be used as a biomarker. Another one that has emerged over the last couple of years, and which was also related, I think, to Loeys syndrome, where maybe early on it was suggested that those patients had something called aortic or arterial tortuosity, which is a twisting and winding of the arteries or the aorta. And that has been used also as a biomarker. The more tortuosity you have, the more twisting and winding there is, the higher the risk for dissection. So here is a study that was done in France in a cohort of patients with TGF-beta receptor 1 or 2 mutations, so patients that have Loeys syndrome. And you can see that aortic tortuosity in itself had some predictive value. For example, here you can see what is called an odds ratio of 2. It means that patients that presented aortic tortuosity had a twofold higher risk of needing a preventive surgery. So that could also be used as a biomarker. The more tortuosity we see over time, the higher the risk becomes for dissection. I also talked about the tissue level and the imaging. There is specific MRIs that can look at the flow patterns in the aorta. Here you can see an aorta on the left, which is quite a normal flow pattern, and then a flow pattern of a patient that has a bicuspid aortic valve. So a normal aortic valve consists of 3 leaflets, but in the bicuspid there is 2. And you can see that the flow patterns are disturbed. They become somewhat vortexed. And studies have been done, and that correlates with how the tissue looks. So at spots where there is high, what they call wall shear stress, so some kind of negative influence on the aortic wall, there you see that there is increased fragmentation of the elastic fibers, and this increased fragmentation in itself is linked to the risk for aneurysm development and dissection. So theoretically, if we could look at this at a tissue level by some kind of imaging technique, it could also help to predict spots where there might be a risk for dissection. Another example of more kind of metabolic study is a study that has been done by Lynn Sakai, who is also very committed to Marfan syndrome, where she's looking at breakdown products of fibrillin molecules. And the more fibrillin breakdown we see, she could associate that with aortic aneurysm. I think she's still studying to see whether we can use it as a diagnostic tool or even to monitor progression. So if we apply the criteria that I outlined, so being simple and fast and cheap and non-invasive and sensitive and specific, you can see that with the current examples, there is none of the biomarkers that have been identified so far that really fulfill all these criteria. And so I think that's why there is ongoing research in order to try to develop a biomarker that is ideal. So, there is many examples at the protein level that could be tested based on different techniques. I won't go into the details. I just want to say some general conclusions on the current studies that looked at proteins as biomarkers for thoracic aortic aneurysm. And then you can see that there's far more studies for abdominal aneurysm than thoracic, and that some of the biomarkers that have been suggested for abdominal aortic aneurysm, which is less genetic and more linked to lifestyle factors, have been also tested in thoracic aortic aneurysm patients. But unfortunately, these are all very small studies that have not been confirmed in larger cohorts. And most of the biomarkers that have been identified so far either suffer from low sensitivity or low specificity. And as you may remember from the previous slides, this is very important, because if it cannot detect the disease by low sensitivity, then it's not a good biomarker. And also what we see is that a lot of the biomarkers have very poor positive predictive value. So they cannot very well distinguish an aneurysm from another vascular condition, which could be an inflammatory disease or a thromboembolic process. They often lead to alterations in the same protein. So then it does not help us to monitor or aortic aneurysm or to predict dissection. So in the ideal world, we would find something that it's easy to quantify in the biological fluid. So this could be either blood or saliva of urine, something that we have easy access to. It would be a cheap test, it would be quick, and it needs to be consistent. And preferably we want to see it increase when the aneurysm is evolving, and increase even further when there is a dissection. And we also want to see that correlation with the progression. So do we think that there is such an ideal biomarker? I don't know. I think we probably have to evolve to a world where we use all different techniques that is now available in biotechnology. We can look at the level of the genes and variation there. We can look at the level of the RNA, which is the transcriptomics. We can look at the level of protein with the metabolomics, which are the breakdown products, so the proteomics, and try by learning from these different applications what good biomarkers would be. And Aline will give an example on how we try to tackle that problem. I think in the future that we will probably evolve to what I call a multifactorial prediction model. So there will not be a single biomarker, but it will be a combination of different risk factors. So here I outlined the ones that we already know. We know that there is genetic risk factors. There is also some dynamic risk factors such as hypertension, and pregnancy could be additional risk. Some treatments could be protective or provoking the aneurysm. But then I think we'll have to add another new category of emerging risk factors and biomarkers, which come from the different categories that we described. And I personally believe that, for example, artificial intelligence will help us to integrate this in a model that will predict better the aortic aneurysm progression or the risk for dissection. So this is the general introduction, and I will now give the word to Aline, who will explain how we developed a specific strategy to identify a biomarker.
Okay, so thank you for the important introduction for the second part. So to kick off, our approach was not specifically tailored to finding biomarkers, but we specifically wanted to look for mechanisms underlying the disease that are shared by different etiologies. So something that is shared by Marfan syndrome patients, but also Loeys syndrome patients, and preferably in the end also by the non-syndromic TAA patients. Because if we can find things that are important for the different subsets, we can raise the attention from the pharmaceutical companies, and we can also offer patient management that is more broadly applicable. So we tried to define a strategy where we could find both biomarkers and drug targets. What is the important difference between both? Where a drug target is something that is critically contributing to the disease process, which is contributing to disease development and progression. A biomarker can be involved in that process, but it doesn't have to be per definition. It can also be something that's innocent, an innocent bystander, as we call it, that's related to the disease process, but is not really driving it. It's something that happens along the way. So we wanted to define a strategy that could find both, because that would give us more information than looking for one of them only. The second thing that we found important is that we wanted to do this in a hypothesis-free manner. So often, such studies are based on what is known already. You pick a couple of proteins from a certain pathway and you look further into them. But now that we have those large omics approaches like transcriptomics, proteomics, we can also do this in a hypothesis-free manner. And if you look at one model specifically, you will have a long list with hits. But if you look for things that are shared between different conditions, the number of hits will be smaller. And this allows us to think outside of the box and to look at a couple of things that are maybe not that straightforward at first sight, but seem very interesting if you go away from what is known already. And then a third aspect that we thought was important was in first instance to stay as close as possible to the site where everything is going on. So to start from affected and control aortic tissue samples instead of immediately going into the blood, because then we can have a more clear idea of what is going on there and afterwards see if it's only also recapitulated in the blood samples. So as you can imagine, and as you know, it's not that straightforward to collect tissue samples from affected individuals, but also not especially from control individuals. So therefore, we decided to start with mice. We do have a couple of mouse models available in the lab. We are familiar with them. It's more easy to study them. You can get results in a shorter time frame. And we decided to focus on 3 specific models that are complementary with respect to their phenotype, but also with respect to the function of the gene that's defective in this specific mouse model. So the first model is a Marfan syndrome mouse model. It's commonly used in the community. It's the cysteine to glycine missense mutation. And this mouse model presents with early onset aneurysm formation, typically at the level of the aortic root, and the distal ascending aorta is involved to a lesser extent. Then the second model is a Loeys syndrome model with a SMAD3 mutation or a knockout. It's in our hands presents with aneurysm somewhat later. So with respect to timing, these 2 are already complementary. Here we also see the aneurysm at the level of the aortic root and less at the level of the ascending aorta. And then the third model is an IPO8 model. So IPO8 is a recessive condition, causes a recessive condition, which is characterized by symptoms that are similar to Marfan syndrome symptoms, but also to Loeys syndrome symptoms. And we also see a bit of overlap with Prince-Goldberg syndrome. Here we have again, an early onset phenotype, but at the level of the distal ascending aorta. So here we see a difference at the spatial or at the region that is affected as compared to the other 2 models. Important is to mention that the first 2 models, they do present with dissection sometimes, but they're rather rare. While the third model, there we see dissections in about 20% of the mice before the age of 32 or after the age of 32 weeks. So as I said, besides the fact that they are complementary with respect to the phenotype, we also see difference in these 3 models with respect to the function that's abrogated. So while we know that TGF-beta signaling is important in all these diseases, we also see more and more evidence that other pathways are also involved and that we should not only focus on TGF-beta signaling, but also on other things. So the proteins that are involved here, like fibrillin-1 is also an extracellular matrix protein, so there might be things going on structurally as well. Then SMAD3 is mostly involved in the TGF-beta signaling pathway, and IPO8 is actually a protein that is involved in the shuttling of cytoplasmic proteins to the nucleus in the cell. And besides the SMAD proteins, there's a wide variety of cargo, so a wide variety of proteins that are shuttled by IPO8. So also here we might expect that different pathways can be involved. So as you notice, we here strongly focus on the syndromic mouse models. Why do we do this? Because the transgenic syndromic mouse models are the ones with the strongest phenotype. So we can start from that. We know them very well. We know their natural disease course. Of course, we would like to validate things also in non-syndromic cases, but this will be done afterwards in the patient study. So how did we approach the hypothesis-free discovery? So for each of the mouse models, we decided to isolate aortic tissue samples, and this at the level of a mild aneurysm, while already early in the disease course. For the identification of a biomarker, you want the aneurysm already to be present to make sure that you can differentiate affected versus non-affected. But if we want to find a drug target, you want to be as early in the disease process as possible, because after a while you also get compensatory mechanisms that are activated, and these are not the things that you want to target. You want to really target the driving mechanisms which happen early in the disease. So this stage is actually a good stage to identify both types of markers that we were looking for, the drug targets and the biomarkers. For each of this model, we determined at which age you would have, on average, the majority of the animals having a mild aneurysm, and we checked with echocardiography that this was true before isolating the tissues, getting RNA out of them and subjecting them to bulk RNA sequencing. So what is bulk RNA sequencing? This means that you just take the entire tissue and you get all the RNA out of it, irrespective of the cell type it's coming from. This has the advantage that it's rather cheap, that it's also not that complex to analyze afterwards, and this gives us the potential to look for preliminary data that our hypotheses are true, that we can find these type of converging mechanisms over the different disease etiologies, that we can find possible biomarkers or possible drug targets, and afterwards which is what we are actually also doing now that we have some preliminary data. We're also focusing on the single cell aspect, because the aorta has different cell types in its structure, all with a different ratio as compared to the other cells. So the vascular smooth muscle cells are most abundant. So the signal that we will get from the bulk RNA sequencing is mostly coming from the vascular smooth muscle cells. But also the other cells can be important. So currently we're also collecting tissues for the single cell sequencing to learn more about the cell-specific effects. But nevertheless, also with the bulk RNA sequencing, we found some exciting results. So after analysis of the RNA sequencing, we found 8 genes that are significantly upregulated or downregulated. So they're significantly more present in the disease or less present in the disease condition as compared to the control mice. And this is shared already between the 3 etiologies. We then decided to prioritize them or rank them in first instance on significance, which means that the effect has to be consistent over the different animals, and also for log fold change, which is actually the strength of the signal. The more upregulated or the more downregulated, the larger the log fold change. Then from the most interesting hits based on this category, we also checked in the literature if any of these genes had already been published to be dysregulated in other aortic transcriptomics data of other mouse models or patients, to get extra evidence that it's broadly applicable. We checked if they were truly expressed in the aorta, which is some kind of technical control as well. And then important, if you want it to be an easy biomarker, as Bart said, it's handy, or it's actually the best, that they are also detectable in blood as the encoded protein. So some of the genes that encode their protein, these proteins are secreted into the bloodstream. And if we can find such a target or such a hit, that would be the most interesting one to follow up on, because then we can just detect it with a blood sample. We also checked in literature if there was already evidence that that protein could be well detected in serum or plasma of control individuals or patients with another indication. Then, as I said, we're looking for new mechanistic insights, but within the group of genes that we identified, we looked if there was something that was maybe not very evidently, but in some way already related to the things that have been described so far. And then we also looked whether the fact that the biomarker that we have in mind, so if one of these hits has already been suggested as a biomarker for other diseases, because as Bart said, then it's becoming very difficult to discriminate between a patient with TAA and that other condition, for example, a cancer patient. So from this prioritization, there are a couple of interesting hits, but there was one top hit that we decided to focus further on. So first, we wanted to validate technically that what we saw in the RNA sequencing experiment is indeed true. And then you have to confirm it with an independent technique. And the technique that we used for that is qPCR. And we really also used a different set of mice. So we bred these mice again, and we used a different colony with the same genetic defects to confirm that what we saw in this initial set of animals is truly the case and that there was not something that went wrong during the experiment. And as you can see here in the graph with the black bars and the white bars, the increase of the potential biomarker was indeed also validated with qPCR in the fibrillin mouse line and in the APO8 mouse line as compared to the healthy controls. Now you also see that there is a gray bar included here, so that deserves a bit of extra introduction. So the APO8 mouse line on a black 6 genetic background, it's a specific mouse background. That specific mouse line presents with thoracic aortic aneurysm early in life. But if we take the APO8 genetic defect and we put it on another genetic background, another mouse strain, the 129 strain, we don't see any aneurysm anymore up till the age of 40 weeks, so rather late in life. So this means that this mouse model has the genetic defect, but does not present with a phenotype, which is actually a good control to see if the biomarker has true biomarker potential, because then we can, if we see that it's elevated in the TAA presenting mouse, but not in the mouse that does not have TAA with the same genetic defect, that is what actually we want to do in the clinic, to discriminate patients that have an aneurysm and patients that are at risk but don't have the aneurysm. So here you can see the data just from the mice that we see an increase in the 129 in the black 6 background in aortic diameter, while we don't see it in the 129 animals. And in the gray bars, you can indeed see that we don't see an increase in our potential biomarker in the 129 strain, which is very reassuring. So then we wanted to follow up further on this. We do that in mouse because the next step is to correlate the biomarker levels with the aortic diameters over life, and also to see whether the biomarker increases at the moment that the dissection occurs. We want to do this in mouse because, yeah, as I said, the experiments are faster, but also you have less confounding factors. We use inbred mouse. So this is a more homogeneous population, and this can give us clearer results on a short term. So we start from the fibrillin 1, the Marfan mouse model, the APO8 mouse model that presents with TAA, and then the APO8 mouse model that does not present with it. We select serum samples through the tail vein every 4 weeks up till the age of 40 weeks. And we always do it from the same animals. And at the same time that we take the serum sample, we also do echocardiography to make sure that we can really compare the aortic diameters that we observe with the protein levels in the serum sample. And then we do an ELISA on it. It's just a technique to quantify the levels of the, oh, we're in the dark. Okay, there's light again. So we can quantify the levels of the protein of interest in a very accurate way. So what will this experiment learn us? It's currently ongoing. It can, in first instance, we want to prove that the aortic aneurysm is truly associated with increased protein levels in serum, because until now we did everything at the aortic tissue level, at the RNA level. So while it is predicted that we would also see that at a protein level in the serum because it concerns a secreted protein, we still have to make sure that it's truly the case. Then secondly, we want to prove that increased secretion is specific for mice with aneurysm. So also when we do the longitudinal study, we don't want to see an increase or a significant increase in the 129 APO8 model that does not present with aneurysms. Then we want to show that progression can be followed and can be demonstrated based on the serum levels. And then finally, as Bart said, something important is also to see if we can detect a peak in the candidate of interest at the moment that there's a dissection or prior to the dissection, because that would also be a very important factor to follow up on in the clinic. Now, mice are very interesting from a research point of view, but of course, we're doing all of this not for the mice, but for the patients. So in the end, we really have to validate everything in a human setting as well. To do so, we collected serum samples also from a substantial group of thoracic aortic aneurysm and dissection patients, as well as age and gender matched controls. We're looking at different subsets of patients. So here we're including syndromic patients, but also non-syndromic patients, patients in which a genetic variant has been found, but also patients that are unexplained, patients with a dissection or patients with a mild aortic aneurysm. So we try to cover different groups that are interesting to follow up on. We take serum from them. The aim was to have more than 200 patients and about 100 controls, and we are above half of it now at the collection, and it goes pretty well. And then also here we want to use an ELISA to determine the protein levels in an accurate way. So here, more or less the same questions can be answered, but then in a human setting. So are the serum levels elevated in patients as compared to healthy controls? Then importantly, is there a difference between subgroups? Like, is it a good biomarker for syndromic patients only, or also for the non-syndromic conditions? And then finally, if this specific protein has been linked to other diseases in literature before and is, for example, suggested as a biomarker for other diseases, how do our concentrations compare between the concentrations that they measured? Can we still discriminate those two conditions from each other, or is it too overlapping, and is it eventually not a good biomarker in the end? And then, so as Bart said, this is our approach to find a novel biomarker or a couple of novel biomarkers, but in the end, most likely it will be the combination of different biomarkers that will give the answer. So this is just a piece of everything that's going on, a piece that might help in finding the ideal subset of markers to follow up on to really be able to discriminate patients at different disease stages throughout life. And then finally, this is more like a lab thing that we're looking into. So we're very interested in doing large scale drug screenings. So seeding cells of patients in our Petri dishes, then adding drug compounds and seeing what does it do on the cells. Do they suddenly become to look as a healthy cell instead of a diseased cell? But if you want to do this in a large scale and high throughput manner, you want a readout, or you want to measure something that's easy to interpret, that is fast. And we think that such a biomarker, and the one that we're now working on, can help here too. So the first question that we want to ask then is, what's the cell type of origin? Because we started with RNA sequencing from the entire aorta. We first have to determine, is it a biomarker, especially as expressed by vascular smooth muscle cells or endothelial cells? And then secondly, if we look at it also in a dish, in an in vitro culture, can we truly discriminate with a good readout the patient versus the control? Is the difference large enough that we can see a significant difference if we add compounds to this? And that is the final slide that we would like to present. This is the Antwerp team, a large group of enthusiastic people that are working together on this project, but also several other projects. And we're happy to answer questions if there would be any questions. Thanks for your attention.
Right now, the only question we have is whether or not this was recorded so people could watch it again. So I was talking briefly with Gretchen McCarrick as well from Dr. Dietz's lab, and she said she was also going to need to watch it a second time. So thank you. There's so much great information here. If you have a question and would like to ask it at this time, feel free to drop it in the Q&A or in the chat. I have both open right now, and yeah, we can see if we can get that answered. Any here we got one that popped up. Are you guys able to see it, or do you want me to? I'll just read it to you. My father recently passed away. Yep.
Oh, no. So I think in genetics, we don't suffer, fortunately, too much from false negatives and false positives. So when a lab does test for a familial mutation or a familial pathogenic variant, then they always take into the experiment a positive control. So you would know for sure that the test worked well and that it does not deliver a false negative result. So when I was describing false positive and false negative, it mostly refers to the detection of, for example, proteins, as Aline described, that you would measure in the blood, and it does not apply to testing of the presence of familial mutations. When you do a first screen, then there could be a false negative, because our techniques to detect genetic alterations are not perfect. So we do know that we do miss things. But in familial testing, the labs take very rigorous measures to avoid false positives or false negatives. So I think you can really trust that test, because those labs are accredited, and one of the key characteristics of an accredited lab is that it can do familial genetic testing with a very high sensitivity and very high specificity. So meaning very low, almost zero occurrence of false negatives. Then the second question is, will this help clarify variants of unknown significance? I think it could. If you have a very, very good biomarker, and your biomarker is very specific for your phenotype, then it could help us to tell that indeed the genetic variation is leading to an aneurysm phenotype.
I also think it more will help the follow-up.
Yeah.
Now you will need to have regular or very regular imaging to make sure that nothing's happening. Then you can just go to your general practitioner, get a blood sample, and then they can check it in the blood sample, which is easier and maybe less of a burden.
Yeah. I don't think we would use it in that way as a first choice. I think it would be more for follow-up and not for I think there's other methods that are better suited to assign a pathogenicity or non-pathogenicity to a variant of unknown significance.
There are a couple questions that have popped up in the chat and here too about time frame for a clear conclusion or when it might be promoted to practitioners. I know it's a bit away from that, but
okay. So first, there's people asking if they can participate. That's a little bit difficult because we process the blood samples immediately, like within 30 minutes. They have to be processed in order to get the different type of samples that we want to do analysis on. So doing that from abroad would require that they locally process the sample, which is not always easy. And maybe I'll let Aline answer the second question on how close this is to clinical practice.
I think it will still take a while because we first need to have the good data in humans. Then you will test it in larger cohorts. You want to see whether, if you do it in larger cohorts, that the specificity and the sensitivity where Bart was talking about is really good enough. And then in the optimal condition, you also want to test, maybe at that stage, you can already see that it's way better than what is available now. But then optimally, you also want to test it in combination with other potential biomarkers to see if you can even get a better predictive value. So yeah, it's difficult, I think, to put a time frame on it. But I would say definitely not in the first 5
to use it everywhere.
years.
No,
Yeah.
I think we could get an answer whether it's worthwhile pursuing it in a larger cohort within a shorter time frame. That's what we're aiming at, to have some very good preliminary evidence to test it then on a larger scale in other centers with more patients. If it's an easy test, it should be implemented quite easily everywhere, so then we can easily get to these larger numbers.
And like also in clinical trials, I think it's easier to implement it there already than just to see how the evolution is of the biomarker upon treatment.
Yeah. So then there is a question about the potential to predict aneurysms at other locations. That's also something that we take into account. We try to gather samples of patients that have no aortic aneurysm at the root or the ascending aorta, but do have an aneurysm elsewhere. And then we can also test whether it has some sensitivity or specificity to detect those. But that will be a separate kind of question, I think, to answer. The first goal is, I think, to find a biomarker for the aortic aneurysms and dissections.
I have time for this one last one here about having a VUS.
Yeah, so I think distinguishing whether a VUS means something or does not mean something is a completely different field, which requires different methods, requires different types of approaches. Most of the time, it would require a test that specifically models that variant in some assay and then see if the outcome of that assay is significantly different from controls and aligns with what we know from pathogenic variants. It's not straightforward to find such assays and tests for every possible gene, but that's definitely something that people are also working on and try to identify.
There's a lot of work being done on developing novel models that are more high throughput as well to test FUSs. So it's also something that's definitely on the scientific agenda.
Yeah, but I think it would be a different webinar.
Well, I would just like to reiterate a couple of the comments that came in through the chat. Just thank you so much for this research and for putting together a really excellent presentation to explain it. A lot of notes about giving hope through this. I know we're early, but even just that, please, I hope you can feel the hope that comes from the communities that would be affected by this, time frame regardless. Obviously we always want it done sooner than later, but I think just knowing that this is being worked on gives a ton of hope to, caregivers and people affected by Loeys disease, Marfan, and then potentially others as well. So thank you both for this time, for the work you've been doing and what's yet to come. And this has been recorded. It will be posted and a link will be shared out to everyone who registered. And then we'll probably also email it broadly as well, because we know there's a lot of interest in this because of that very hope that you're providing to so many. So thank you both, and have a good rest of your evening.
Thank you
For you as well.
Bye-bye.
Bye.